Hi everyone, so we're going to discuss the lab on chemical equilibrium and Le Chatelier's principle in Chem 10. And the goal of this lab is really to observe Le Chatelier's principle where we're able to shift chemical reactions either towards the product side or the reactant side depending on what we do to it. As a reminder, there's two different types of reactions that we observe. The one that we're going to be interested in here is a group of reactions that are called reversible reactions, where the reaction will go towards products, as you might expect when you have a lot of reactants. But then what happens is some of these products can then revert back and form the reactants again. And so that ability to go back to form the reactants is what makes it a reversible reaction. There's some other reactions where the movement is primarily one way, so we would refer to those as irreversible reactions. We use this double arrow symbol to indicate that that reaction is reversible. Now, the problem with reversible reaction is you might think that it will never really end because you never really run out of reactants. So for an irreversible reaction, it's pretty easy to see that the reaction will be over when one of your reactants is out, right? Your limiting reactant in that case. But for a reversible reaction, how do we know that it's over or it's complete? Well, the way we know is when the reaction reaches something called chemical equilibrium, which is the point at which the rate or the speed of the formation of the product is exactly the same as the speed of the formation of the reactant. So in other words, the rate at which the products are formed is equal to the rate at which the reactants are formed. So at that point, you basically don't get an excess over time of either reactants or products, and so we say the reaction has reached equilibrium. Once a reaction is at equilibrium, you can shift the reaction towards the reactant side or to the product side by disturbing or stressing or perturbing the chemical system. This stress or perturbation can be of different types, as we discussed in lecture. We can either change the concentration of a reactant or a product by increasing or decreasing it. We can change the volume. And if you're talking about aqueous species, that's typically done by evaporating the solvent or adding more solvent. If you're talking about gaseous reaction, usually you're changing the size of the container, making it bigger or smaller. You can also change temperature, and temperature has effects that depend on the type of reaction from an energy perspective, whether it's exothermic or endothermic. Now, you can also add substances that have no effect on the equilibrium. So these are things like catalysts, inert substances like nitrogen gas, for example, or helium gas. So those things don't really have any effect on the equilibrium itself. This idea that the reaction will shift to the right or left, depending on the type of stress or perturbation that you add to the reaction, that's what we call Le Chatelier's principle. And so we talked about these in lecture, but I do want to give you an example here to help you answer the questions that are going to be relevant for this lab. So the questions with this lab, a lot of it has to do with starting with a particular reaction and then making sure that reaction has reached equilibrium and then doing something to the reaction at equilibrium and seeing which way it shifts towards. Okay, so here's an example. So you have a reversible reaction between two gases, NO2 and N2O4. This reaction is fairly easy to see in terms of which way it's going because NO2 has a brown color while N2O4 is colorless. So if you see in your container you have mostly a brown color, then you know that you have a majority of NO2 gas. If you see mostly a colorless appearance, then you have mostly N2O4 gas. It says, consider the observation below and answer the following questions, right? So the system that you're supposed to look at is this system right here. So we start originally with a tube here on the left where it's filled with a gas. Maybe this picture is not too clear, but this water is warm and then this water has ice in it. So you move it to the ice water. But it tells you here as well in the question, what do you observe when a sample of NO2 gas is placed in the beaker containing warm water on the left compared to ice water on the left. What I notice is that when it's in the warm water, it has a very brown color, and then when it's in the ice water, it has a much less intense brown color. So that tells me that in here we have a lot of the NO2 gas, and in here we have a lot less of the NO2 gas. In fact, we have more of the N2O4 gas, which is the colorless gas. So that's the first observation. So you can say the color of the gas is less brown as a result of that movement. And then the second question is, in which direction did this stress cause the equilibrium system to shift, and explain that? Well, clearly we're going from more NO2 to less NO2. So in other words, we're shifting to the right in this case. What's causing the shift in this case? Because the color is associated with NO2. If you're becoming less brown, that means you have less NO2 in the container. Now, what factor causes the shift? Temperature, obviously, because it's the change in temperature. We didn't do anything else, right? Now, the next question is this an exo or endothermic reaction? Explain. Okay, now, as I said earlier, temperature effect depends on the type of reaction you have. So in this case, we're shifting to the right when we decrease temperature. We can look at this table to see what is the type of reaction that will result in that kind of a shift. So shift to the right, decreasing temperature, that tells you that it's an exothermic reaction. The explanation we use in class for understanding this type of shift is, remember that an exothermic reaction means that heat or energy is released by the reaction. So you can consider energy to be a product. Why is decreasing temperature result in a shift to the right? Well, decreasing temperature is kind of removing energy or removing heat. So if you remove product, then the reaction should shift to the right to compensate for the loss in product. Okay, so that's the kind of analysis that you have to do for the questions in this lab. You have to answer a number of these type of questions, again, with both observations as well as explanation. Right, so I'll mention briefly the different reactions that you have to work on. There are 5 different reactions. The first one is just a reaction, or if you want, a physical system where sodium chloride in the solid form is dissolved in water and forms the ion form, which you can't really see because that's the aqueous form, right? And so the question here is, once it reaches that equilibrium, your experiment will require you to add HCl, and then you would try to determine whether it shifts to the left or to the right. Now, of course, if it shifts to the left, you'll be seeing solids form. If it shifts to the right, these are aqueous species, which means they're transparent to you. The second one is a chromate solution, and as you can see here, it's a chromate plus H+, which is proton or acids, and then it forms dichromate and water. Now, the easy way to observe this is by its color. So one is yellow and the other one is orange. I'll just point out here that sometimes people tend to not quite know what the color that they're looking at. So you just have to think about, is it more yellow? Is it more orange? Okay, that's really very similar to this. The way I interpret this earlier is that I see one is brown, the other one's less brown. Okay, so same idea here. You just have to think about which one is more yellow or less yellow, and that would sort of tell you which way it's shifting. In this case, you're going to be doing 2 steps to it. You're going to first add HNO3, which is an acid, and then afterwards you're going to add to that same test tube NaOH to see which way it shifts. So you have to record the shifts for each of the addition. The other one is an ammonia system, which looks like this, and it has a color because you've added an indicator in there. You've added phenolphthalein. So when NH3 is present, then it's going to be clear, whereas when OH- is present, it's going to be pink. You're going to add a solid to this and then mix it. The solid is ammonium chloride, and you want to see which way it shifts to. And again, all you have to do is to see, is it pink or is it less pink, right? Because that makes it clear which way it's shifting. Then you have this cobalt chloride system, which is something we refer to as a complex ion. You don't need to know that too much at this point, but the idea is that there's 2 different forms of these ions, and one of them is pink, the other one is blue. You might not think of them as very similar colors, but they're sort of in the same region of the electromagnetic spectrum, so sometimes people tend to get confused by that. So again, just think about, is it closer to a pink color or is it closer to a blue color? And that's how you determine whether you have more of this species or more of that species. You're going to actually prepare 3 different samples. The first sample you're not going to do anything with, so that's your control sample. And then the second sample, you're actually going to add HCl, and you're going to try to see it shifts more more towards the pink side or the blue side. And then the third one, you're going to add ammonium chloride, and again trying to see if it shifts more towards the pink side or the blue side. To that third one, once you add the ammonium chloride, you're also going to do a temperature change. So you're going to heat it up and then cool it down. So very similar to what was done to this sample right here. You're going to put it in warm water and then afterwards in ice water. And you try to see as you do those things whether it's shifting towards the blue side or the pink side. The last system is this iron thiocyanate complex. Again, we're going to know which way it's shifting by looking at the color of the species that we see in the test tube. So one is yellow and the other one is red. The deep red is very red, it's blood red, so that usually it's fairly easy to see. In this case, you're going to have 4 different test tubes. The first one you're not going to do anything with, that just shows you the control. The second one you're going to add some iron chloride to it and then see which way it shifts. Third one you're going to add some potassium thiocyanate, again seeing which way it shifts. And then the fourth one you're going to add some silver nitrate. Now that one is a little interesting. Hopefully you can see that silver or nitrate is not in this reaction, but you'll see some kind of a shift, and what your goal is to try to explain why you see that particular type of shift. Last point here is just to point out that this reaction or experiment does have a number of things that are fairly toxic, so you want to be careful. When you use 12 molar HCl, you want to be very, very careful because it's corrosive, and if it falls on your skin, it would give you acid burn immediately. So just be very careful and use it in the fume hood. When you heat the test tube in part 4, also make sure that the heating, you don't just put your test tube straight on the Bunsen burner like that and not move it around, because when you do that, what happens is that a localized part of the test tube gets heated at very high heat, and then that generates a huge bubble of gas, and that bubble of gas is gonna pressurize the solution and pushes it out of the test tube. And that liquid, of course, can splash on you, can splash on other people around you. We call this bumping. So to prevent overheating and bumping, what you do is you move the test tube back and forth on the flame of the Bunsen burner. And that would break up that large bubble of gas into smaller bubbles, which makes it a little less likely to do something like this. The last part of this is to make sure that all the waste that you have in this experiment is discarded in the chemical waste container, which we have in the fume hood.